welcome everybody to the Researcher Roundtable series hosted by the AD Data Initiative. And today we're going to be talking about a cool topic, imaging or innovative imaging modalities, I should say. And we're really grateful for our panelists and our moderator that have agreed to share their wisdom with us today. So thank you guys for being here. A couple reminders for our audience before we get started. First off, you can ask questions throughout the event. You can just head over to the Q&A icon that should be in the top navigation bar there in Microsoft Teams and ask our panelists any questions you have. And if we don't answer it throughout the discussion, we'll make sure to get to those in the dedicated Q&A session at the end. Also, reminder that you can not turn your cameras or your microphones on. Only the panelists and the host will be able to do that. So the Q&A will be the best option for you to ask your questions. And finally, this event will be recorded and posted on AD Connect. So I think that's all from me. I'll go ahead and stop sharing my screen here and hand it over to you, Natalia, if that's all right for intros. Thanks.
Thank you so much, Monica. Good afternoon, good morning, good evening to everyone. Many thanks to all the attendees. I think this is a very nice research roundtable about a very nice topic, innovative topic. We're going to talk about innovation in imaging modalities and with an amazing group of panelists. My name, I think maybe some of you know me already, but my name is Natalia Vilor-Tejedor. I'm a team leader in genetic epidemiology and biostatistics at the Barcelona Brain Research Centre, and I will be moderating this session. I want to really thank Frederick, Michael, Barbara, and Singh for your time to be here today, because as I was saying, I think this is a very important topic. I want to also thank the ADDI for organizing this session. And maybe for starting, and also for those people in the audience who may not be familiar with the field, I will ask you, seeing Michael, Frederick, Barbara, to maybe introduce yourself a bit, also to provide a quick background of your work, especially focusing on how brain imaging plays into your research. So maybe, Michael, if you want to start.
Sure. Thank you very much, Natalia. Thanks for having me. It's a great pleasure and honor, obviously, to be part of this distinguished panel. My name is Michael Scholl. I am a professor at the University of Gothenburg and also have a part-time position as the principal research fellow at UCL, at the Dementia Research Center there. I have a background in PET imaging. I was lucky enough to do my PhD under the supervision of Agneta Nordberg at the Karolinska Institute back in the day. So she was the first to do amyloid PET scans. It's quite exciting to grow up there academically. And I was then also fortunate enough to do my postdoc with Bill Jaggers at UC Berkeley, where we worked on some of the earliest tau PET studies. I am now a professor here in Gothenburg, and my group focuses basically on a combination of biomarkers, both imaging-based and fluid-based, and reaching basically all the way to digital biomarkers to help identify neurodegenerative diseases as early as possible.
Thanks, Michael. Maybe Singh?
Yeah, hi everyone. Thanks, Natalia. I'm Sean Dione. I'm a senior program officer at the Bill Melinda Gates Foundation, and as a technical lead of our neurodevelopment and neuroimaging work there. And although that predominantly focuses at the other end of the age spectrum, so early childhood and maternal health, before I was at the foundation, I ran a large longitudinal study lab at Brown University, where we really focused on using MRI to look at brain development and brain change across the lifespan, from fetal through to aging and into dementia. So again, looking sort of womb to tomb, as it were, and really trying to understand how the brain changes, both in healthy development, healthy aging, but also in various conditions, be it psychiatric, neurodevelopmental, or neurological disorders throughout the lifespan.
Thank you. Maybe Barbara?
Thanks, Natalia. And thank you again to ADDI for this interesting discussion and also for inviting me. So my name is Barbara Marebwa. I am a staff scientist at the Michael J. Fox Foundation for Parkinson's Research. So my portfolio is basically imaging development and validation. So as we're trying to find imaging biomarkers for Parkinson's disease and of course other related synucleinopathies as well. So I am looking forward to this discussion, and thank you so much for the kind invitation.
Thanks, Barbara. And finally, Professor Marco.
Hi, Natalia. Yeah, thanks for inviting me to this session. Also, thank you to the ADDI to share some experience with it. I'm a neuroradiologist, and so I grew up with MR. And like Sean, I've been intrigued by using MR as a tool to study brain development and more the end-of-life changes that we will be discussing today. So to do some work with in pediatric neuroradiology, leukodystrophies, but also in midlife, MS and white matter diseases. But then I have a keen interest in late life changes, so white matter changes that occur with aging, but also brain aging in general, normal, pathological, and how do you disentangle them, and using images to extract endophenotypes that will allow you to study the various disease processes as they unfold, and people, going towards Alzheimer's disease as my particular interest, of course, and that we can complement it with PET imaging, as Michael already mentioned, but also looking at other diseases, looking at atrophy patterns, looking at disease heterogeneity, and using that sort of endophenotype to understand, whether different diseases or even within one disease, whether there are multiple trajectories and why are they there, and try to link it to genetics, to other biomarkers, and to try to predict outcomes and treatment-related changes.
Thank you so much to all of you again for being here today. Actually, we are here for talking about brain imaging. And well, as you know, brain imaging has been considered in the neurological field as a key variable or variables to understand more complex brain processes and diseases. So maybe one first question is, in your opinion, how has brain imaging contributed to what we know about Alzheimer's disease today? Who wants to be first? Maybe Sim, if you want.
Sure, I can take a crack at that, and then be expanded upon and corrected by the fellow people on the table. But I think, certainly it'd be hard to argue that modalities other than MRI have done a tremendous job of really advancing the field. In particular, I think Michael was mentioning all the PET work going on to, again, be a little bit more specific and sensitive to the traditional hallmarks of the disease, right? So both the tau pathology and amyloid pathology, and I think has really led the way on that specificity. Certainly, I think, the work in MRI in being able to accurately track and follow longitudinal progression of the disease, looking at atrophy, some of that early work on those longitudinal studies showing atrophy within the hippocampus, for example, as well as throughout the brain, was beautiful work. Again, just showing that these changes are happening well in advance of very, very profound cognitive deficits. And so I think, and then that obviously is beginning to progress as MR becomes a little bit more specific and sensitive with different modalities, be it, looking at more magnetization transfer or MT or other diffusion measures or mild water imaging to look at a little bit more sensitive microstructural change, but certainly not getting to the specificity of the pathology that PET or other imaging modalities will give you for any means, by any stretch. But I definitely think in looking at the overall pathology and pathological progression of the disease, I think imaging has made a pretty substantial contribution. Whether that contributes into biomarkers or targetable biomarkers is, I think, a bigger and open question. But certainly, I think understanding the disease progression, I think there's been a tremendous work. But yeah, happy to hand over. I'm sure Michael can lax lyrically on the role of PET and whatnot, and certainly Freddie on other bits.
Yeah, I don't want to be the pure PET guy here. Obviously, I acknowledge the contributions of other imaging modalities, and I think we'll revisit those later during the debate for sure. But obviously, when it comes to Alzheimer's disease, I think the combination of imaging-based biomarkers really has told us in vivo, is the only in vivo evidence that shows us that there are several subsequent disease phases that start with amyloidosis, that followed by tauopathy, And we can then assess neurodegeneration and actually have measures that actually also correlate and associate with cognitive decline. So if we just focus on these really core processes that we believe underlie Alzheimer's disease, I think we have an incredible portfolio at our hands now that has taught us what we partially knew from autopsy studies, but has added this way of sampling the brain now, instead of having these small cubes that, you know, a pathologist might look at maybe and then find there's a tangle, there's a plaque, but we sample the brain now at a, you know, at best probably a 4 or 5 milliliter cube voxel basis. And I think that is really the revolution, that we can sample the entire brain, assess it for proteinopathies, neurodegenerative processes, and have this combination at our hands now. So I think it's been an absolute major contribution to our understanding of AD. It has taught us a little bit about what we don't see also, that there might be co-pathologies. I mean, there are definitely cases where the full picture of what we have at our hands in terms of modalities doesn't tell the whole story. So I think that's also a learning from having these imaging modalities at hand. Yeah, and I think also, what I mentioned before, really this relationship between what we've thought we knew from autopsy or postmortem studies and what we see in vivo, I think there's still a lot to learn, but it gave us a hint towards what might actually work in a living brain rather than the deceased one. Yeah, I'll hand over.
Yeah, and I agree, and I think, you know, if you make a comparison with pathology, I love to work with pathologists and do MRI pathology correlations. But, you know, pathologists also envy us because, you know, they have, you know, the final verdict, but only at the end of the disease, mostly, you know, occasionally with a biopsy. And then they have very detailed information, but only for a very small part of the brain, because they cannot sample the whole brain under the microscope. So, you know, they have the microscope. We have what I call the macroscope, because we can see the whole brain. And not only can we see the whole brain, we can look at it repeatedly. So we can, you know, look from the first symptoms or even before the first symptoms and do repeated measurements. So it's really a complementary thing if you compare it to pathology, and most pathology agree with that. You know, they're very interested in seeing the imaging data in earlier stages and looking at the whole brain. And when I started working in this field, Alzheimer's was, you know, a clinical diagnosis, which was, you know, very often wrong. And then even looking at something simple as hippocampal atrophy was really a game changer. And also when I started using MRI in clinical trials, you know, the first cholinesterase inhibitor trials in MCI, and we started doing imaging, it was scary what you would see, what people would call MCI. There were people with beautiful normal brains, there were people with devastated brains, there were people with enormous vascular co-pathology, people without, so it was massively heterogeneous. And I think imaging helped to sort of frame the population better, and obviously with the more molecular specific information that, you know, we've been even more specific, but still it's scary how heterogeneous populations can be if you just look at the clinical label.
Maybe continuing to this point, I think it's very interesting, all of this explosion of technology. Do you think that all of this amount of data, heterogeneity that you are commenting, have harmed somehow our progress in Alzheimer's disease? Like, for instance, by giving us false conclusions or misleading some evidence? What do you think about this?
Well, you know, for some parts, not at all. For example, you know, if you have a positive amyloid scan, you know, there's no doubt about it. You know, you have amyloid. Whether you then have Alzheimer's disease is another matter. But, you know, if you also have a positive tau scan, so that is super specific. I think, you know, what we've learned is that, for example, amyloid can occur, you know, years before people have what we call Alzheimer's dementia. And it can be co-pathology, of course. I think it's a bit more complex with MR, where you have if you just focus on atrophy, you have neurodegeneration. Well, neurodegeneration is just an end product of many processes. If it occurs in a specific region, then you have some specificity. For example, if you have severe left-sided temporal lobe atrophy, you have semantic dementia. You know, I see them every week in our clinic, and you know, if I see that picture, I'm never wrong. If you have hippocampal atrophy, becomes slightly less specific, but, you know, good chances that you have AD. If you see generalized atrophy, you know, it could be lots of things. But in real life, people have multiple diseases, of course. They don't just have Alzheimer's disease. They also have hypertension. They may have another disease. But you see a sort of more convoluted picture.
Yeah, I'd say sort of taking on to that, the imaging itself is just a tool. It's our interpretation that I think leads us into these sort of, murky waters, as it were, right? So, we might have our favorite. I'll speak to MR, given that's my bigger knowledge. And again, it lacks that sort of specificity because we're not labeling something in particular, right? In essence, we have a really big device that images water predominantly, unless you're doing spectroscopy or something. And water can be clever and tricky, and we can try to be clever and tricky in how we image it, but we're actually just imaging water. So, what it's telling us and how we're interpreting that, I think, is where we get into a little bit of trouble. If we think of some of the metrics and look at how they've been looked at across the time span. When we had diffusion tensor imaging come out, fractional anisotropy, that was clearly myelin, right? That was all that was. Well, except you have fractional anisotropy, pretty high levels of FA, even as an infant without a myelinated brain, right? So surely that wasn't quite as specific as we thought it was going to be. And so all these little changes, I think, again, kind of linking back to what Dr. Barkov was saying, is these things are composite measures of multiple underlying pathologies that are happening, and we just choose one thing that we're quite good at and say, well, this is clearly what we're looking at, right? And it's not quite that clear cut. And so I think that's where the interpretation gets in. The tool itself is just doing exactly what we ask it to, right? It's producing an image. It's doing exactly what we want it to. And I think it is contributing to our knowledge. It's just how we interpret it. Very much like, and I think it kind of follows, and I can't remember the author, and so I apologize for that, but there's a beautiful old sort of paper called How a Physicist Builds a Radio, or How a Biologist Builds a Radio, which sort of follows, the biological perspective of how you would look at it and reverse engineer a radio, which I think is almost how physicists try to learn neuroscience, actually. But again, it's the interpretation that's always the challenge.
Yeah, and just to add to that, of course, the tool tells us what it tells us, but I think we've also had a bit of a reproducibility and replicability challenge in imaging in our field, right? And I think these are because of reasons that we are aware of, and I think we're now making concerted efforts to address, right? So when I was in school, if you had a data set of 80 people, that was considered, a large data set, and you can't really generalize anything from 80 people, right? So having consortia such as ADDI coming together with a lot of data that is representative of, a large variety of populations can help in things like that. And then when you think about, having different protocols in different sites, interscanner variability, etc., I think these are challenges that we have had in the field, but we are making concerted efforts to, to help with, data harmonization, things like Combat that you can do post-processing or even before you do the reconstruction of your data. So I don't think all in all we have stifled the progress of Alzheimer's disease. There have been technical challenges and interpretational challenges that we have had that I think we are on the way to getting on top of.
I do want to add one aspect, though, where I feel like it definitely has not stifled our progress, but it has probably made us partially blind as we keep staring at the same aspects of the disease because we have the techniques and the modalities for those. And I think we've been probably using them slightly lazily because we're really using them to describe what's going on. So we keep describing the trajectories from the modalities we have, which sometimes makes us forget, what else could be going on and lead to the enormous heterogeneity. But we're not really using them yet to describe mechanisms in the way we probably should. That's a feeling I have increasingly over the past years. Of course, this is how you start using a modality, but maybe I'm probably not smart enough to do it, but I'm sure there are people who could use these techniques more smartly than what's been done so far. So I think the not forgetting that we only stare at the things we can actually measure, because as you said before, Sean, the modality is doing what you tell it to do. But then there is a way of using them smarter, both when it comes to the scientific hypotheses behind it, but also not dumbing down imaging modalities to a level of a fluid biomarker, right? Why are we binarizing every single PET measure? I mean, the entire strength of a PET measurement is the spatial information, right? And that we can quantify regionally different pathological burden. PET is a quantifiable modality. We shouldn't forget that if you use it correctly. So there's a lot of information taken from this that is very often discarded. And I think this is a simplistic problem sometimes, and it makes interpretation not complete. So it's, as you all said, not the modalities per se. It's probably how we use them that might have stifled some of the understanding.
Thank you, because I think you are all raising very interesting points. And maybe perhaps moving to a more clinical perspective, because now we know about the potential of AD biomarkers in this era, but also I'm curious about the possibility of prescribing therapies based on imaging results. I think it's a tricky, this question, but do you think this is a feasible way to treat Alzheimer's disease? Feel free to jump.
Well, it would be prophetic to say yes, right? I mean, eventually we will get there. So you could say yes. I think again, it kind of comes back and it builds on this exact conversation we're just having in terms of how are you interpreting that, and then what treatment do you fall back on? For example, if you were to see a change in MRI measured functional connectivity, what the heck do you change to change that, right? What does that mean, right? You would never use that as a guiding tool in terms of intervention or of treatment, because it means all sorts of things, right? So again, I think it comes back to how do we understand the data that we have, and do we understand the progression of the disease well enough to then where that biomarker fits into that disease pathogenesis to actually begin to make a treatment decision based on it. I think we have a ways to go, but ultimately, I think with enough work, we will get there. But if I were to go see my neurologist today and they said we're going to put you on this drug because of an MR finding, I would look for another neurologist, I think. So yeah.
Or for a better radiologist, yeah.
Yeah, well, either one, yeah.
Listen, but I think I think the goal is not to treat images. So that's not the goal, but they're an adjunct to select the right people for treatment. For example, think of people with Alzheimer's disease. You would like to make sure that they have amyloid on board and not start an antibody as they did in the papanosumab studies in people who were, I think, up to 30% didn't have amyloid pathology. That's not a logical strategy. But in some instances, if you look at the AHEAD 3-4-5 studies, that really try to prevent very early Alzheimer changes, there the goal is actually to find the amyloid in the brain, confirm it, and to stop it from progressing. So then you're very close to where imaging is really the target, because it shows you the primary pathology that you're looking for. But another more typical clinical scenario, of course, it's an adjunct to make sure that you're treating the right patient with the right drug, and then preferably at the right time, with not too much or too little tau pathology, for example, making sure they're not at risk for side effects. So, to prevent them from developing ARIA, for example, people with microbleeds, safety monitoring, but also efficacy monitoring. It's super interesting to see that with amyloid, but also with anti-tau drugs, you can actually remove the protein of interest, and then perhaps at some stage we will be able to titrate the drug to make sure that people fall below a certain level and then monitor them. But that will be in the future. Let's say 10 years from now, perhaps.
I think we need to make sure what these surrogate endpoints really mean, right? And then probably apply them on a rather individual level. Because, if you have someone early with early amyloidosis, then amyloid is a valid surrogate endpoint if you remove it. But if someone already has the brain full of tau, then amyloid is not a valid surrogate endpoint, because if you remove the amyloid in this patient, then probably whatever tau is triggered in terms of downstream, or the downstream mechanisms we believe come after amyloidosis have done too much damage. So I think it's a little too simple to say, treat them based on an amyloid PET scan, but I think a combination of imaging modalities might very well be the way to prescribe therapeutics if the endpoints are validated. So I think Frederick, the MS field has been quite good at that, for example. And I had this discussion with my colleague Nico Franzmeier yesterday, who really talked about this again. I think it's maybe there's not a one fit for all strategy when it comes to treating based on just one modality, or certainly not.
Yeah, well, thank you very much for everything that you are sharing, because it makes my brain is thinking a lot right now, and also I'm thinking about these last 5-10 years, how much we advance in the imaging field. And if I try to, let's say, summarize or think about the major things that we got during these last years, actually my brain is plenty of things, but I want to know your opinion. What do you think is the most exciting advancement that we got with in the imaging brain field? It's difficult to choose one, I know, but maybe you have to spot something. What is your?
We can only choose one, Natalia. We can only choose one each.
You can choose two, but yeah, let's say just one, because otherwise I think we can here spend a lot of time. But yeah, I mean, of course, if it's two things that are equally important for you, you can spot two, of course.
Well, I'll go different ends of the spectrum, right? So for me, I'll start with maybe ultra high field MRI. So there's been a lot of efforts to make them safe. So not only for basic research, but now even for clinical diagnosis. There are a lot of centers in the US and in Europe that do have this ultra high field MRI. And of course, it's going to help to reduce, the gains on SNR are going to allow these fine structures to be visualized, detect all these microstructural changes that we think maybe occur before all these mesoscale atrophy that occur, whenever you have clinical symptoms. So maybe that may also assist in molecular imaging technologies such as MRS and CEST, which are specific for certain metabolites. And I know there are a lot of drug trials that are going to be specific for maybe inflammation or mitochondrial challenges, etc. And then on the other end of the spectrum, I'm also going to talk about, low field. Maybe Sean has a lot to say about this, but yeah. Low field and portable MRIs. I think that's also really, really exciting for me, especially in resource limited settings. For example, this is basically democratizing MRI, right? You don't need to build a Faraday cage or whatever to have people do MRI. And then, of course, with advancements in AI, we can borrow from deep learning reconstruction techniques and have patients not for 1 hour, but maybe for 5 minutes, and you get everything you need by undersampling case space. Borrow from computer vision and super resolution and make those low field images high field, so that you just don't use it for looking at stroke and tumors, for example. You can now start looking at hippocampal atrophy and microstructural changes that may be important for Alzheimer's disease. So I'll stop there. Those are my two extremes that I think have been very exciting for me in the past 5-10 years.
Yeah, I'll jump on, because I totally agree with the low field and high field work. And I think, the low field work is really just getting going. And I think right now it's focused on trying to make those low field images look like a high field image without actually exploiting the physics of low field, right? Which could actually tell us quite a bit that we can't do at high field. And the fact that we're actually even being able to now think about doing even like lactate imaging, potentially even doubly labeled water to look at almost an FDG type PET image at low field without any sort of radio tracer or whatnot. The ability to get them anywhere. So when we were at Brown, we had one in a Ford Transit van that we could drive around almost to anywhere. There's a group in Arizona that has one in the back of a trailer that they're just driving around Arizona right? So the idea of, not having a study population, a clinical population for the research side that's majority, white, high income, male typically, but getting into more diverse populations to understand the disease. But then also when we come time for treatment, being able to put these devices almost anywhere so that people having these more advanced treatments that might need further follow-up or more invasive follow-up have access to that at their convenience, right? They don't have to go to a high-end imaging center. So I think there's a tremendous amount that's going to happen around that. That's going to really sort of open up our understanding of what's going on. And it's weird to think that, the first MR scan was done at 50 millitesla. So the idea of having a 60 millitesla scanner now is not crazy, right? We just have to go back and remember what all those pioneers from the 20s through to the 70s were playing with, right? And figure out how they did stuff and how we can kind of replicate that. So I think there's a lot of interesting work there. And again, as I think as Barbara was mentioning, we can go anywhere, right? We have them in sub-Saharan Africa, Southern Asia, where you just couldn't put a high field system. So again, they're not a replacement by any stretch, but I think they are a really cool additional tool to help understand things, and again, help when we get to the treatment aspect. Because then you can also do huge populations, right? The scan costs on a low field system is about $50 a scan, right? Compared to the $1,000 I would pay at Brown to use their major imaging center. So if I can automatically increase my patient populations by a factor of 20 without increasing the cost of the study, that's huge, right? I'm either putting bigger populations in, or I'm scanning them more frequently. There's a huge amount of information that we can learn. So I'm super keen and super excited about where that field
clearly enthusiastic, and I share this partly with you, Sean. Although it's being hyped a little bit, people keep talking about portable scanners, and in fact, you know very well they're not portable at all. You can move them around, you can wheel them around if you're lucky within a building. I think the name portable is a bit of a euphemism.
Well, I mean, we've put them in a van, we've put them in trailers that you can move around and take to like an assisted living center. So I think they're at least more portable than a 1.5.
They're transportable,
but, yeah, but they're not an ultrasound system or something on your phone. That's for sure, right? So they're definitely not pick up in a briefcase or a laptop and carry with you, for sure, absolutely.
No, because I've also seen people said, no, we should do a project and use this portable MR, and we go to people and scan them at their home. Well, it's not that simple, I think. Yeah, but now there are lots of interesting developments, and I think that's the fascinating thing about imaging, that there are so many new techniques coming around. Obviously, the molecular imaging techniques have revolutionized the field, the amyloid, the tau PET tracers, but also newer tracers coming along almost every day for inflammation, for receptor density, you name it. But also to come back to MR, MR also keeps amazing you all the time with the new techniques that come along and that mature. Think of ASL, for example, that is getting much more mature. But then you have neuromelanin mapping, susceptibility weighted imaging to look at iron, and think of all the developments in network imaging. We can look at the functional and the structural network of the brain in many ways and try to understand how a complex organ like the brain works with crude approaches by looking at centrality and hubs, etc. But that is getting increasingly sophisticated. And if you then think of the various types of networks that we can look at, we're just starting a project now to understand how these various types of networks actually are interlinked, and how a functional and a structural network, how are they interlinked and how do they influence one another. So we're just scratching the surface of what is possible, I think, with imaging.
Me, clearly, tau PET, number one. I think that has been the most important imaging modality for Alzheimer's disease in the past 5 to 10 years. But I want to move from MRI also a little bit and make a case for CT, because if we talk about accessibility and availability, CT really is the workhorse, and it's completely forgotten. It's not hot, nobody really cares about it, but it's done routinely in many countries, and it's also just way more available in many parts of the world. And here I think photon counting CT scanners are really exciting development, because you can reduce radiation massively, you can reduce scan time massively. So it's a little bit like low field MRI, but actually improve image quality, but with much reduced burden. So I think that is a really interesting development that I think can have great impact on just, routine scanning that is readily done. But I think CT really is a bit of a forgotten modality that has a lot of potential, because it is routinely not really used the way we use MRI, even if it's just about volumetric measures. It's usually just visually assessed, and very oftentimes it's only just assessed to exclude non-degenerative causes of say, cognitive decline. But I think there are now AI-based solutions that help also reliably derive volumetric information from standard routine CT scans, and I think that can really be a nice addition to what's readily acquired in clinical care already. I think those to me are two most important developments.
Can I add something to this, Natalia?
Sure.
Because CT had an interesting history. Was sort of mature, saturated technique until the multi-slice CT came along, and you could, rather than when I was a resident, a CT was, you made one slice, you had to wait 1 minute for the image to reconstruct, you had to move the table, do the next slide. And then you had, after half an hour, you had a 15-slice CT scan where you could barely see anything. Nowadays, with the multi-slice, you just flow through the gantry and you have, 300 slices in 30 seconds. And I think, and that brought it back for a lot of applications, especially outside the brain. But I think now there's a bit of a silent revolution with MR occurring, with the compressed sensing and the AI-based acceleration stuff, where you can massively undersample case space and get, a very good 3D T1 in under 1 second. An any type T1 you can get in 1 minute, I'm sorry, and multiple other contrast. And so this opens the door to doing much bigger, much more extensive studies with multi-contrast imaging, doing, speaking to Sean's quality, doing a good quality T1 or T2 mapping in just minutes, rather than, remember, Sean, the 20-minute protocols with, the couple of days of reconstruction. All of these things can now be done on the fly, so that's super interesting. And also for PET, of course, Michael, you yourself have been experimenting with lower dose PET scans. I think AI will help us to speed up things massively.
Yeah, exactly. I remember back in the days when, you shot for like 15 minutes because that wasn't suddenly clinically feasible, right? Which is not at all clinically feasible. But yeah, now getting into the minute, 2 minute, 3 minute range, which is actually now clinically feasible, I think.
Well, it seems that these last years were very exciting for brain imaging. But what about the future? What promising imaging applications or innovations do you see?
That's a question for Barbara, I think.
Well, I am a bit of an optimist, and I'm going to go back to artificial intelligence. I think the application of AI, Dr. Barkov already mentioned a few of them, right? Helping us undersample case space, and especially for Parkinson's disease, where you don't want to keep your patient in the scanner for, 20 minutes or so, you can get this data really, really easily. I think that is a huge promise. And then even beyond that, thinking about technical challenges that we have in MRI, right? So like reconstruction, like harmonizing data across different sites, etc. I think AI, is promising in that it can help us overcome a lot of these technical challenges. I think imaging by itself, of course, is also growing exponentially, where now we can have molecular MRI type of images, the improvement of cells, as long as you have like this exchangeable proton, you can basically measure anything, right? Well, within reason, but certain metabolites that are important for, Parkinson's disease or Alzheimer's disease, other tauopathies, etc. So I think these are the ways in which in the next 5 or 10 years, AI, for example, is going to be very useful in imaging.
Yeah, I would
there's
also
actually a
add,
question in the Q&A around this as well. Huh? So I agree, Barbara, it's super interesting what is happening in terms of just doing faster and better acquisition techniques, noise suppression, artifact removal, etc. But also, once you have the images, try to extract, even the information that we want to know now, doing better brain segmentation, but also to extract new types of information that you wouldn't have necessarily thought of. That is, a vibrant area of research, I would say.
yeah, for sure. I completely agree.
Also,
I think that the AI is such a cool idea. I'll also put a plug in for canonical AI, our intelligence, right? As we slowly get more and more smart about what we actually want to image and the ability to then develop acquisition techniques around that. So, not only just improving the reconstruction of the type of information, but improving the information that we're getting out of the data. And I think that then leads into, I think a big revolution coming in terms of combining these modalities, which we've not traditionally been awesome at, right? In terms of fusing the information and taking information across large data sets, different modalities, all telling us complementary and sometimes, different but complementary information, and how you bring that together. And I think, as we advance in this world of both high-end equipment, lower democratized or more accessible equipment, to get greater information, how we begin to fuse that information is going to be a really powerful but tricky problem. So I think that's where I'm excited to watch how that expands as well.
Yeah, and then in terms of large models, like LLMs did millions of parameters. I think ImageNet had 1 million or so images to be able to train it. Probably for us, we won't be able to get to 1 million or so, but having consortia like this come together, pull their data, and then of course also using things like synthetic data generation. I don't know what you feel about that, generating synthetic data to help train the AI as well. So augmenting the data sets that we already have, I think is also going to be interesting. Things like active learning, we do need a lot of label data sets, right, to be able to train these AI models properly. We probably don't have doctors to label our images, but if you use something like active learning, where you label a subset of it, you give it to the AI and it tells you, oh, this confused me, and then you label it and you continue training it progressively, then the AI can go ahead and label the rest of your images as well. So in terms of generating all the huge amounts of data that we need, in terms of making sure that the data is ready for the AI, so that we give it high quality data, so that we get a high quality output out of it as well.
Yeah, I really think AI is going to be the way to make existing modalities a lot more available, a lot more informative. And then the parallel development will likely be to have novel ligands on the PET side, and novel modalities like super high field and ultra low field MRI, new CT technologies, CT. But when it comes to PET, Felix mentioned this before, I really think here we have to work incredibly on accessibility and availability. I think this is the modality that is stifled and limited most by that. And it doesn't have to be that way. I think we are picturing PET as this incredibly exotic and expensive and complex modality. And sure, you need a cyclotron, so that's a bit of a downside, obviously, compared to an MRI or a CT scan. But once you have that, and you have an established tracer already, chemistry that has a routine in production, then it doesn't have to be that hard. I think we are using it the way it's been used since the 1970s, basically, late 70s. And why do we, for example, inject these enormous doses of pharmaceuticals, of radiopharmaceuticals? It's just not necessary. And there's now enough papers out on FDG, for example, and we've tried that for amyloid, and we're trying it for tau ligands. There's enough evidence that you really don't need that. So you can scan a lot more people with one batch. That's just the protection side of things. But then there's the ethical part when it comes to research, right? That is incredibly limited by that you can only scan individuals 1 or 2 times. And at least in Europe, depending on the country you live in, your ethics are going to be very, very strict on how many scans you can actually administer. And also here, you can, think of PET studies that can all of a sudden do 10, 15, 20 longitudinal scans. And just imagine the incredible information you can derive from studies like that. And it is perfectly feasible. I think we just have to be a bit braver in how we're using the existing technologies, both leveraging AI also to interpret, again, existing modality data, but also the reduced resolution and dose data we can actually derive at lower cost and at lower burden. So to me, this is a near future development that I think can really revolutionize the way we use our imaging modalities. Readily.
Thank you all for your vision. I also see that to move forward in the field, we must really join forces. And this also means joining knowledge and data. And talking about data sharing, I think I would like to ask you this because I think it's very important. How might data sharing help or maybe being detrimental for progress in this field, in the field of brain imaging, for understanding better AD, also Parkinson? What is your opinion about data sharing?
Should be mandatory.
Yeah, I totally
yeah, and
agree.
I should really, that sounds like a joke, but should be mandatory. It's such a waste of time to collect data and then to just keep it to yourself. Because at least I'm very limited in what I can think of, what I can do with my data, and I'm sure when I have my data and I give it to Michael, he will have bright other ideas, and if I give it to Barbara, she will have yet another bright idea to do with it. And then if you give it to another person, they would have other ideas with it yet. And so I'm personally curating 2 big imaging data sets from EPAD and from AMIPAD, and they're massive data sets, not just the imaging, but they have GWAS data, they have endless risk factor data, clinical data alongside, and the number of possibilities of things you can do with the data sets are almost endless. And certainly something that I cannot do myself, but even with a large group of collaborators, we cannot exhaust the possibilities. And this must be true for other data sets as well. So I'm very much in favor of it.
Can I just play devil's advocate for a second here? Because obviously I completely agree with everything you say, Felix, and I also think data should be shared. But what are the incentives for clinical scientists to actually conduct these kind of trials? I mean, we all know, and most of us have done it. It's incredibly time-consuming and nerve-wracking to be a scientist and conduct these, even single-site studies, but even more so multi-site studies. The bureaucracy is absolutely deadening. There's very little support oftentimes from the institutions. And all of a sudden, you're there spending these grants that you've gotten over the years, and you had to jump through all these hoops and get ethical approvals and talk to the medical product agencies and get them to approve a couple of PET scans. I mean, we've all been there, right? So what's the incentive for someone to do that and then readily share everything? And and there's people who build perfectly fine careers on these existing data sets. I completely agree.
But there are
I
multiple
really don't
ways
want to be
share
cited
data.
that I don't want to.
You can throw it. You can just throw it over the hedge, but you can also share it and enter. Lure people into a collaboration with you and work with you together. Which I often see happening because lots of people don't understand the data well, so they like to engage with you and work with you, make the best of the data. And if you look from the funder perspective, if let's say I know of a lot of big population studies that are ongoing, and they're good studies, but then the government spent tens of millions to do these studies. And only a limited number of people work with it, and sure they cannot exhaust the possibilities of what you could do with the data. So from a funder perspective, you could argue that data shouldn't be kept by one particular researcher. Although I fully agree, there's a lot of work that goes into it, and this should be acknowledged. But that can be acknowledged with group ownership, like ethnic-styled group ownership or otherwise.
Yeah, I completely
don't get
agree.
me wrong. I completely agree. I know we agree, but it is just
for argument's
I
sake.
think the incentive is a big challenge, right? And particularly, you think now the way that a number of the large, at least NIH studies have shifted. You know, you look at ECHO now, which is a big early childhood, early environmental influences on childhood development. HBCD, so another large longitudinal study of neuroimaging in kiddos, the Connectome projects and whatnot. Those are almost all now funded just as data acquisition studies. There's very little funding in those mechanisms for actually data analysis. The goal is just to make it available, which I don't think is the right incentive, right? You're just basically paying people to go and collect data when these are really great scientists who can also bring that knowledge to bear on the data analysis. So I think, you know, certainly that building in that incentive to share that data, and I think we're building that capacity now as we've gotten closer to this, right? I remember back, you know, if we were having this conversation probably 3 years ago, the biggest complaint would have been, well, I won't get tenure because I'm making my data available. I don't get credit for it. I think that's kind of been, you know, I think that's still a valid point, but I think it's becoming less so as people get more comfortable with the idea of these large multi-site studies. I think now the next thing is how, again, do you continue that credit along, and whether that's, again, moving more towards an open science approach where you have almost even like a blockchain type, you know, audit trail of who was involved in the data, and your name and your lab and your efforts are always sort of, you know, tracked along and acknowledged at each stage. You know, whether that's the one way, whether it's, you know, direct data sharing, or we move into like a federated learning type approach where you're not actually just dumping the data into a single archive, but people have access to it and people can still work with each other. Because ultimately, I think the biggest challenge that you have is, as you mentioned, Michael, people have made really great careers out of analyzing data in big repositories. But there's a lot, as we move to larger studies, and particularly multi-country studies, the context in which that data was acquired is going to become more important. And the only people that have that knowledge are the people who acquired it, right? So, you know, that local contextual knowledge as to why you might be seeing different things. And so I think there needs to be almost a rethink of how the governance of these large studies are actually conducted, and how the data is actually shared. I think the existing model of either just chucking it into an archive is probably not going to be sustainable in a way going forward. I think there has to be some other bits around there, again, around that context, or else, you know, how the heck do you compare a data set from India with a data set from Indiana? Right? Like, how do you do that? There's such different contexts, and you have no idea what the various environmental factors are. So yeah, I think we almost need to be really thinking about this, and what that governance and how we're actually going to share that data is going to be done.
Yeah, I'll just, oh, go ahead, sorry.
No, I was just going to say, and that's even after you get the fact that India won't share their data, right, as a government. So even after you solve the governmental issues and the politics around data sharing. But sorry, Barbara, go ahead.
No. I completely agree with everything that has been said. And I just mentioned, as a funder, so we fund a lot of studies, and now we actually do have an open access policy in place that we put in your contract that says that if we fund you, then, you know, the data, the code, etc. must be made publicly available, right, in open repositories. But I think we are lucky in imaging in that we already have standards kind of that make this a bit easier, right? We have BIDs, we have IDM. So there are a lot of very smart people working on ways to make sure that we can share this data in a standardized way so that it's useful for everybody, right? So that it's not just throwing data into a repository, but, you know, describing it with a data dictionary so that the next person who comes in knows what you did, right? Versioning the data, for example, so that it's reproducible, etc. So I think in imaging, we're a bit lucky because there's a lot of thought that has already gone into creating these standards, making it easier to share, reproduce, etc. We just need to have a bit of a culture shift, right? And make sure that, you know, researchers are buying into, oh, sharing my data is actually a positive. And we are doing it with the carrot of, oh, this is the money, but you have to make sure that you share the data as well. So I guess it's just trying to convince investigators, and of course, telling them that there are ways to make this easier for you, right? There's tools that you can use to make this easier for you as well.
Yeah, thanks for sharing the views, because I know that the sharing generates a lot of controversy. But actually, I want to raise also another point, which is, and I was discussing also with the ADDI team, at the end, data sharing is, let's say, promoting a more, let's say, balanced and equitable research environment, no? Because at the end, in some places, there's no budget or way to get access to this data. So this is all, at the end, promoting diversity. And is promoting also to provide the same opportunities to everyone around the world to make questions and use the data for answering these questions. And I know about the controversy and about what Michael was saying, but also I think there's so many positive things, not apart from for the global research, but also for promoting diversity. And I think I would like to thank the ADDI initiative and also the contributors, because I think this is a very important point. And maybe we have time for last question, because there's some questions in the chat as well. So maybe very, very briefly, I would say, how can we improve the access to images also? Apart of data sharing, do you think that there's a way of improving the access for like primary but also secondary analysis? What is your experience about this?
I mean, as a funder, my experience has been everyone wants to share data until they have to share data. That generally seems to be the way it is. Everyone's keen when they're writing the grant, and then when they actually have to do it, suddenly there's 1,000 barriers that suddenly come into place. And so this is a question that we think about a lot. And I think, when you talk, I think we're well versed in the global north in particular. I think maybe even Europe is a little bit ahead of the US in some ways, with GDPR and being really defined as to what can be shared, when it can be shared, et cetera. I think a challenge that we obviously have is when you start talking about diversity, you move into a number of global south areas. They simply don't have the connectivity to transfer some of these large data sets, right? Some of the areas that the foundation works in, we're really working on 3G cellular networks. So sending a gig data set is going to take hours. Right, it's just not something that can be done on an easy framework. And so the solutions the foundation has taken is around things like Flywheel, which is a platform that allows data curation, but also harmonized data analysis, which is another aspect, right? It's one thing to share all this data, but then you might have to reanalyze it. And if you have a million data sets, to reanalyze it every single time is going to be challenging. And maybe that's another area where AI sort of begins to play a role into making these analysis steps a little bit quicker. But I think that's another thing to think about is how do you actually get this data into a usable format that can be shifted around very, very easily, not just when you have, gig Ethernet fiber, but you have a 4G cell phone. And so thinking about that is going to be a bit of a challenge, but that's something that we need to address as well. And then you have, as I say, the various political issues where, certain countries make data sharing completely impossible. And so you need to work with just within the confines of, say, or the confines of federated learning type approaches and other machine learning approaches that don't require the raw data, but can train on other aspects or keep things separately. And so, we talk about distributed power. Maybe that's the next way, right? Is that it actually is distributed network that you're trying to learn from.
But I think another thing to consider is, and I've seen that, for example, with the EPAD data, which have been collected with the purpose to share, and it's been in the ADDI platform for quite some time, the tabulated data, but we've also now provided access to the imaging data, so people can request the imaging data. But then the requests are often poorly formulated. People just say, we ask what type of data do you want? Well, send us everything. And then so we get into contact with them. Yeah, because we wanna develop an AI algorithm to differentiate AD and FTD. Well, the whole study is not about FTD in the first place. So people just click on a button and then they just say, please send me all your data. Well, without having a clue what the data is. And you could argue, well, perhaps we didn't specify very well, or you have to ask them better questions. But that's the other thing. If you want to broker between people who want to do research and people who have data, you know, you need to tell well what data you have, but people also need to think and ask the right questions, if you see what I mean. I don't know whether other people have similar
sitting on the
experiences.
review panel of Amipath, Felix. Yeah, I really think what Sean said before is so important, because on top of the repositories and the infrastructure, it is about making the infrastructure rather universal, because it has to work in very different legal systems. And being European and being part of NIH-funded projects sometimes is an absolute, it's a huge problem, right? GDPR, you mentioned this, Sean, as something positive. I tend to not agree with that. I think it has been an absolute nightmare, honestly, because it is a complex framework that a lot of lawyers can interpret individually. So we're very often in the hands of an individual lawyer who can tell us what we can share and what we can't share. So that is a nightmare if you have collected large data sets, obviously. And they, you know, there's this whole debate about what is pseudonymized versus what is anonymized at this point. So really, I feel at this point, rather than the technical challenges and even harmonization challenges, it's the legal challenges that are at this point stifling data sharing when it comes to neuroimaging data, at least here, as the most challenging. And I don't really know how to overcome this, because there is a legal framework, obviously, but maybe funders and certain research councils could work a bit more concentrated or an orchestrated effort to make sure that there are policies in place where we as researchers could refer to and say, look, this has been done, this is a precedent, it's legal, it's been done in Europe, it's been between the US and India and Europe, you name it. So this would help enormously, because we have, as you said, you know, people want to share, and then when they see what it entails, you can spend hundreds of hours, you know, fighting your own lawyers, and they tell you, you know, pseudonymized data is not shareable, and everything is pseudonymized data if you dig deep enough. So that is a realistic big problem.
Yeah, that is true. Much like what we talked about initially of imaging and stifling biomarker discovery, it was interpretation. And I think in this case, the stifling of data sharing is the lawyer interpretation, which is always a challenge, that's for sure. Yeah.
So
Yeah,
maybe
no, lawyers are there to confuse us. But we also confuse the lawyers. I've learned over the years, you know, what you have to do if you go to a lawyer, you have to give them a bit of context. Not just send them a protocol and a lengthy document and let them then start from scratch and start thinking about possible dangers and scenarios and IP and what type of law should apply. I think if you enter in a discussion with them and explain a little bit more the context and the real risks, then it becomes better. And it's similar with ethical committees, you know. Ethics is super subjective, and every ethical committee will have a different opinion. And sometimes you just need to go into a discussion with them.
Thank you all. I'm gonna go very quick to the chat because there's a lot of questions. I'm gonna probably have time to read a couple of them. So the first one says, what does the near future looks like for dissecting the cerebrovascular component of AD and other dementias?
That is a very good question. Close to my heart, if I may. Yeah, so vascular is very diverse, you know, very different types of vascular pathology. You know, you have hyperperfusion that you can measure. We have white matter hyperintensities, of course, which are a popular topic, but there are also vascular components which we don't understand very well, such as the cortical microinfarcts, you know, where we know there are hundreds or thousands of them. We don't see them very well. It's only the tip of the iceberg. So I would love to have a better, and then there are lots of vascular mechanisms like hypoperfusion, you know, microemboli, atherosclerosis, arteriolosclerosis, vascular reactivity. There's so many things that can be altered in the vascular system, which, you know, will affect the brain itself or also affect the course and the unfolding of Alzheimer's disease, for example.
Firstly, I would like to see a little mechanistic link between amyloidosis and vascular changes, obviously. So I think we really need a good biomarker for CAA, something that helps us decipher what's CAA and what's not CAA amyloidosis in the brain. So I think that would be really interesting in the vascular aspect of AD.
Yeah, because the microbleeds are clearly also the top of the iceberg only, yeah?
So maybe just the last question that is related to something that mentioned Barbara before. How do you use artificial intelligence, computer vision, and data analysis to enhance the interpretation and visualization of brain imaging data? And what are the potential risks and benefits of these techniques?
Yeah, so at the foundation, I hope most of you know about PPMI data. So this is our flagship study we've been supporting since 2010. And as you know, from 2010 to now, there have been improvements in imaging, in MRI, in the quality of the data that we've been collecting. So one way we are using AI computer vision, for example, is through super resolution, right? So just trying to improve the quality of the data that we've been collecting from 2010 so that it's useful for us now in 2023. And it does seem to work really well, especially in the small microstructures of the brain, which are important for Parkinson's and Alzheimer's disease, right? It makes it easier to segment, for example. And these are, I think, one of the areas where we had this reproducibility challenge because everybody had their different ways of segmenting the STN or the LC, for example. And maybe we need an automated way of doing that, right? So again, there are a lot of AI methodologies or solutions that are helpful in doing things like that, for example, right? So that is just one example in which we're using AI to do that. The other way, apart from the technical challenges, is actually to get information from the images, right? So as I said, I think we will need a lot of images to train so that you can understand maybe there are microstructural changes that occur really, really early on that, for example, maybe a deep learning CNN can help you detect, right? So you will need a lot of these data sets. And I think it's also important to include different tauopathies, different nucleopathies. So as we've been talking here, there's been a lot of talk about, metabolic changes. We have that in Parkinson's disease. You have it in Alzheimer's. You have it in LBD, for example. So is it mostly like a kind of pattern analysis type of challenge, right? I think Dr. Barkov said, if you have atrophy in this part of the brain, I can probably say this is the type of dementia you have, right? So that seems like an interesting problem, an interesting pattern recognition problem for an AI to solve, for example, right? So you tell it this pattern of atrophy, for example, is specific to this type of dementia, and this pattern is specific to another type of dementia, and then let it do that work for you, right? So these are a few ways in which I think AI might be useful in this field, and especially since we now have enough data that we can start building interesting models to answer some of the questions that we have in the field.
Yeah, thanks, Barbara, and thank you all. I don't know if anyone wants to add anything else, because I think we don't have time for more questions.
Yeah, I think that's a really good use in terms of image quality transfer from older scans to bring them up. I think, as I say, other possibilities around that are really trying to leverage how little data do we actually need to acquire. I mean, if you think back, people in radio astronomy have been doing this. They basically get one arc in K-space, right? As the Earth zips around the solar system, we get one line in K-space, as it were, and they're able to reconstruct beautiful images. And that's been done for years and years, even before the advent of AI. So thinking through, like, how much data do we actually need? If someone from radio astronomy looks at us, they're like, wow, you guys get reams of data. What are you complaining about? So I think there's a lot of areas in there in terms of improving, or maximizing the information content from images and speeding up improvement. I think that always comes with the risk of, are you actually then seeing the pathology you think you're seeing, right? Or are you glossing over it? Or are you just making the image look normal, right, based on the training data set. And so I think that's always going to be a little bit of a challenge to think through and make sure that we're not just conforming to the model, as it were. That model is generalizable. But and then, as I say, I think there's a lot to be done in terms of, even on a simpler scale of taking all that data and figuring out how to fuse it and how to extract meaningful data from it. And that's everything from the imaging data to all the contextual information that comes along with that, right? If we have patient reports, particularly ones that have filled in answers by the patient as to their symptoms or whatnot, trying to condense that into a meaningful covariate, I think is another area that it can be used very rapidly, and probably things like ChatGPT could be brought to play right now, right, and sort of consolidating that or bringing information from that. So I think the sky's the limit, really, as to how we think about using it. I think if anything, we're probably thinking the easy low-hanging fruit right now, and haven't really thought through the implications that it will bring.
Yeah, well, thank you all again, Frederick, Michael, Barbara, Singh. I think it's time to finish. I would like to spend hours here knowing your opinion about a lot of things, maybe in another occasion. So thank you again for being here and for your time. And I will give now the floor to the ADDI team for closing this session. Thank you.
Yeah, thank you
very
already
much.
so much, Natalia. Wonderful job moderating. To our audience, I posted a link here in the Q&A chat. If you have any lingering questions or thoughts, or maybe you have a thought after having some time to digest everything that was discussed today, you can head over to AD Connect's Community Discussion Forum and connect with our panelists and moderator there. And also look out for the recording that I'll be posting on AD Connect very soon. So thank you all again. Have a great rest of your day.
Thank
Bye,
you.
everyone.
Thank you. Bye-bye.
Thank you. Bye-bye.
Thanks for organizing.